Hey everyone, this is Dr. Mark Hahn, and this video is going to review the histology slides, specifically the epithelium that I have included in your lab resources on Canvas. So remember, epithelium is one of 4 of the tissues that we are covering in histology. The 4 tissues that we're covering include epithelium, connective tissue, muscle tissue, and nervous tissue. So a little thing about epithelium, remember this is the type of tissue that forms sheets, it forms linings. When we are going over epithelium or when you are labeling epithelium for your practical exam, remember you can use the abbreviation EPI, EPI for epithelium. Okay, so let's take a look at our different types of epithelium with the slides that I've provided for you. So this first slide, remember when you are looking at your slides, you have to take into account the location of your tissue. This is a slide showing you kidney tissue. Now, there is actually 2 types of epithelium that we can see on this slide. Now, remember when we identify or classify epithelium, we look for 2 things. First, we look at the number of layers. If it's 1 layer, then of course it's a simple epithelium. If it's many layers, we call it stratified. Then we look for the cell shape at the apical surface of the epithelium, and we have our 3 shapes. We have our flat or disc or pierogi shape or ravioli shape. That is a squamous epithelium. A cube-shaped epithelium, we say it's cuboidal. And if we have cells that are taller than they are wide, we say that it is a columnar type epithelium. So here we have our kidney slide, and there are 2 types of epithelium that are represented. The first type of epithelium, which is the simplest of our epithelium, is a single layer of flat-shaped cells. So here in the kidney, we have a structure known as the renal corpuscle, which is part of the nephron. We know that the kidney, of course, filters blood to form urine, and the nephron is that main functional unit of the kidney that allows the kidney to filter the blood to form urine. And 2 structures that make up the nephron are the renal corpuscle and the renal tubules. The renal corpuscle is made up of 2 subparts. That includes the inner glomerulus. We said it looks like sort of a little puffball or a cluster of capillaries that bring in the blood to filter the blood to form urine. So surrounding this little puffball is a single layer of squamous-shaped cells. So this is our simple squamous epithelium that makes up what's known as the glomerular capsule or the Bowman's capsule. So simple squamous epithelium represented in our kidney slide. Surrounding the glomerulus and the glomerular capsule or Bowman's capsule, we see a simple cuboidal epithelium. Now sometimes they kind of look like donuts. So here's a donut, here's a donut, but as you can see, our renal tubules making up or made up with another type of epithelium, just a single layer of cube-shaped cells, and that is our simple cuboidal epithelium. Okay, so definitely 2 types of epithelium that we see on this slide. On this kidney slide, we have our simple squamous epithelium, and our simple cuboidal epithelium. Okay. The next slide is actually showing you a sample from the lung. So we said that the lungs are important for exchange of our respiratory gases, our oxygen and our carbon dioxide. And our simplest of all of our epithelium, the simple squamous epithelium, is the epithelium that does that function. So remember, on the practical exam, you will be asked not just to identify the epithelium, but you may be asked to identify a function of that epithelium. We know simple squamous epithelium, important for not only filtration, as we saw in the kidneys, but of course the exchange of our respiratory gases. So this is actually a slide of the lungs, but it is at a more lower magnification, probably either at 5x or low magnification. But we can start to see the alveoli. So the alveoli are the sacs within the lungs that allow for gas exchange. When we breathe in oxygen, the oxygen goes through the simple squamous epithelium to be picked up by our red blood cells, and then the red blood cells give off carbon dioxide to diffuse through the simple squamous epithelium so that we can exhale out that carbon dioxide. So here we're starting to see these alveolar sacs or alveoli, and they of course are lined with a simple squamous epithelium. This slide is just another kidney slide that you guys can review. If you need to remember how to make your PowerPoint flashcards, please check out one of my videos that explains how to make PowerPoint flashcards. But again, you can see the simple squamous epithelium surrounding that puffball or glomerulus. And then you see your little donuts in the periphery made up of the simple cuboidal epithelium. There's the lumen in the middle, and then a single layer of cube-shaped cells. Okay, so again, you can see the simple squamous epithelium here surrounding the glomerulus, and then the simple cuboidal epithelium here making up the renal tubules within the kidney. The function of our simple cuboidal epithelium, of course, is for secretion as well as absorption. We know that the renal tubules are important for helping to form urine. Basically, secreting the stuff that we don't want from our blood and absorbing the stuff that we do. So we secrete out waste products such as urea and other things, and we absorb things that we need such as water, important ions, glucose, all the things that the body needs. So only about, of all the blood that is being filtered by our kidneys, only about 1% actually forms urine. So we do a lot of absorption of the things that we need, and only 1% gets secreted out and forms urine. Here is just a zoomed in picture at a higher magnification of our lung slide. Again, our alveoli are those spaces lined by our simple squamous epithelium, allowing for the diffusion of our respiratory gases. So this is just a lung slide, and again, we see our simple squamous epithelium. Here is just another look at the kidney slide. This is more of a longitudinal view of our renal tubules. More than likely, this is within the medulla or the inner layer of the kidney, but as you can see, we have our single layer of cube-shaped cells forming our simple cuboidal epithelium. Again, this is found within the kidney. More kidney slides. Look for your little donuts here. There's the lumen or space within the kidney tubules, and donuts being lined with our simple cuboidal epithelium. I say they're donuts just because they look like donuts. These are actually the renal tubules within the kidney. Say donuts, lifesavers, however you like to perceive them, but again, made up of a simple cuboidal epithelium. Moving on to our next type of epithelium. Here we see a single layer. So again, we are going to say that if it's a single layer, then we classify it as a simple epithelium. We see cells that are taller than they are wide. So that means we have a simple columnar epithelium. So simple columnar epithelium, you'll see the nuclei kind of arranged in a nice little row, usually towards the basal surface of the epithelium, but it may look a little confusing because we see multiple nuclei, but for the most part, it's usually how the tissue is cut. Look for very neat rows of nuclei. Not many of them reach the top. They're actually just all in a nice parallel row. Among our simple columnar epithelium, you'll see these, they're actually glands, unicellular glands, but we call them goblet cells. So goblet cells are important because they secrete mucus to the surface of the epithelium. So you might see this type of epithelium along the important structures, important organs of the digestive system, such as the stomach. You might even see it also in our intestines, the small and large intestines. Simple columnar epithelium, important for such functions as secretion as well as absorption. We know that the goblet cells do secrete mucus on the surface of the epithelium. You especially see this in the stomach because the stomach is highly acidic. And then in the intestines, you might see small extensions at the apical surface of the simple columnar epithelium known as microvilli. And these are important for creating surface area at the top of the epithelium so that we create more surface area to allow for absorption of nutrients. We know the intestines, specifically the small intestine, is important for the absorption of our small molecules of food. In the large intestine, we know the simple cuboidal or simple columnar epithelium, important for the majority where the our water is absorbed. So simple columnar epithelium mostly found in our digestive system organs such as the stomach and the intestines. And again, you may see some goblet cells as well as the microvilli at the apical surface of the simple columnar epithelium. Here is a slide showing you another type of epithelium. Again, we see not one but many layers of cells. So here's the apical surface, here's the basal surface, lots of layers. So we classify this type of epithelium as a stratified epithelium. We then look at the cell shape at the apical surface, and it is a flat shaped cell, so we classify this as a stratified squamous epithelium. But we have to further identify it as either non-keratinized or keratinized. Non-keratinized means that this epithelium does not contain the protein keratin, whereas keratinized epithelium means that the cells within the layers of the epithelium do contain the protein keratin. I know that this is a non-keratinized stratified squamous epithelium because I don't see distinct layers. Non-keratinized stratified squamous epithelium is actually found within the mucous membranes within our body. For example, we have the oral cavity. Keratinized, sorry guys. Non-keratinized stratified squamous epithelium. So our mouths, our oral cavity, lined with a non-keratinized stratified squamous epithelium, the esophagus, usually areas that are subject to a lot of abrasion within the body. We know the uterus, for example, the vagina, the anus, all lined with a non-keratinized stratified squamous epithelium. We all know what happens when we eat hot soup, or I like to use the example of Captain Crunch cereal or Crunch Berries. We know that messes up our mouths. And it's this non-keratinized stratified squamous epithelium that allows the epithelium to heal itself, because we know that the cells at the basal surface are mitotically active, meaning they're always constantly undergoing reproduction, as well as basically repairing and replacing the cells at the more superficial apical layer. So the basal cells of this epithelium are constantly undergoing mitosis. So non-keratinized stratified squamous epithelium, again, found within the body, specifically our mucous membranes, such as the oral cavity and the esophagus. Now, of course, we have an epithelium that might not appear like the other epithelium. Here is the apical surface. We know it opens up to a free lumen. Might look a little strange to you because it's on its side, but this type of epithelium is special because, as you can see, we have the nuclei at many different levels, giving off a fake stratified appearance. So we say that this is a pseudostratified columnar epithelium because pseudo means fake, but we know the cells are actually columnar cells that all attach to the basement membrane. And it's just that not all cells can reach the surface, but it's still considered one layer. And again, the nuclei at many different levels giving off that fake stratified appearance. Now, at the apical surface, we see these structures known as cilia. So we say that this is a ciliated pseudostratified columnar epithelium. We find the ciliated pseudostratified columnar epithelium, especially in our trachea slide, but this type of epithelium mostly found in the upper respiratory tract. This is also a type of epithelium that lines the upper respiratory tract because it helps to protect the body. We have our goblet cells, which you can kind of see between our epithelial cells that also secrete mucus, so that the mucus will line the surface of the ciliated pseudostratified columnar epithelium, and any particulates such as dust, pollen, allergens, bacteria, anything that enters our nasal passages or respiratory passageways can then be caught in that mucus, and the cilia can then move this material and help us to expectorate out any of the particulate that might be caught in the mucus to also help protect the body. So this is just a slide at the lower magnification. This is our ciliated pseudostratified columnar epithelium, but we find this type of epithelium again in the upper respiratory passageways, especially the trachea. So I know this is trachea because to the far lower right of the slide, there's my hyaline cartilage that actually makes up part of my trachea. We know it's made up of these cartilaginous C rings that again are made up of a hyaline cartilage. So here we see another epithelium that actually has many layers. So we say this is stratified. I know that the apical cell shape is a squamous shape. So this is another stratified squamous epithelium, but instead we see a keratinized stratified squamous epithelium. I know this is keratinized because I can see my distinct layers. We find keratinized stratified squamous epithelium, of course, in the dermis of the skin, which is the most superficial layer of the skin. So this whole layer is the epidermis, which we talked about, or we will talk about in the slides covering the skin. The epidermis is made up of distinct layers. We know that we have the epidermis of thick and thin skin. This, of course, is the epidermis of thick skin because of the thickness of the most superficial layer, which is the stratum corneum. But as you can see, we have distinct layers. We have our stratum basale, our stratum spinosum, stratum granulosum. Above that, a beautiful clear layer only found in thick skin, which is the stratum lucidum, and then the most superficial layer, which is the stratum corneum. So the epidermis made up of a specific tissue. If you see a slide and it's asking you to identify the tissue in the field of view, this is a keratinized stratified squamous epithelium. Specifically, we find this slide. This is the slide showing you thick skin. The thin skin slide, most likely the scalp, will not have that stratum lucidum because stratum lucidum only found in thick skin. Comparing it to our other stratified epithelium, which is our non-keratinized stratified squamous epithelium, where we do not see those distinct layers, but we see many layers of cells, with the cells at the apical surface being a squamous-shaped cell. So this is a non-keratinized stratified squamous epithelium. So you can see, comparing it to keratinized, keratinized has those distinct layers versus non-keratinized, no distinct layers. This is just another slide showing you a higher magnification of our simple columnar epithelium. Look at those nuclei all in lovely little rows. You can see a little bit of microvilli at the apical surface. We have our beautiful goblet cells that will secrete mucus onto the surface of the epithelium. Just another view of our ciliated pseudostratified columnar epithelium. Again, you can see those nuclei at many different levels, giving you that fake stratified appearance. In a sideways view, this time showing you a simple columnar epithelium. Again, simple columnar, you're going to have a nice parallel row of your nuclei. You will see some goblet cells also secreting mucus to the surface of the epithelium. And at a lower magnification, just a beautiful slide showing you your simple columnar epithelium with those beautiful goblet cells in between. And just another view of your ciliated pseudostratified. I'll spell it out for you guys. Pseudostratified columnar epithelium. All right, don't mind the doctor handwriting. But don't forget, we do see cilia at the apical surface, so this is a ciliated pseudostratified columnar epithelium. This is one of my favorite slides, just because it's stained a lovely dark pink, almost purple. So the epithelium here is upside down, but if it's asking you identify the tissue at the end of pointer, this would be a single layer of cells that are taller than they are wide. So this is our simple columnar epithelium. It's upside down because I know this is the lumen right here, that space. That space where the apical surface opens up. So simple columnar epithelium, beautiful goblet cells you can see opening up to the apical surface. And look at the nuclei just line up in a row. And the beautiful columnar cells that are taller than they are wide. So we have simple columnar epithelium. This showing you a beautiful example of our non-keratinized stratified squamous epithelium. Again, many layers of cells with the squamous shaped cell at the apical surface. However, we do not see distinct layers, so this is a non-keratinized stratified squamous epithelium. Oh, this is our last slide. So the last type of epithelium that we will be covering in the practical exam, we call this a transitional epithelium. Oops. The slide keeps disappearing. There you go. Transitional epithelium. So this is called transitional epithelium because the epithelium actually transitions from one appearance to another. We find transitional epithelium in urinary system organs such as the urinary bladder, the ureters, and parts of the urethra. So these organs hold urine. The transitional epithelium basically accommodates the amount of urine that is in these organs. So, for example, if we have an empty urinary bladder, the epithelium is going to take on what kind of looks like a stratified cuboidal appearance. We have these beautiful puffy dome-shaped cells at the apical surface. We see many layers, but if you have a full urinary bladder, what happens is this epithelium will then stretch to accommodate the amount of urine that's in the urinary bladder, taking on a stratified squamous epithelial appearance. But once that urine has been voided or taken out of the body, it then returns back to its puffy dome-shaped stratified cuboidal appearance. So it transitions from one epithelial view to another. So that's why we call it transitional epithelium. All right, guys, so that is the epithelium that you will expect to see on the first practical exam. Again, please make sure you review these slides as well as the resources that I provided for you. Make sure you are working on your spelling. And then take a look at the other histology review video that is on my YouTube channel. Like and subscribe. And please enjoy these slides. Good luck and study hard.